Hello everyone and welcome back to Neuropsych. So in the previous video, we went over a little intro to spatial behavior. We talked about the four subspaces or the four types of space, and we went over five examples of topographic disorientation. We also talked about how topographic disorientation can occur in retrograde and also in anterograde, and we talked about the dorsal and ventral streams in the brain. So in this video, we're going to talk about four types of spatial behavior and some experiments to measure that performance or that spatial performance. Okay, so all animals, including us, have evolved remarkably in our range of behavior in relation to space. So non-human animals now migrate through routes. They can secure locations and nest there. They can find locations of food sources and of places they have cached their food, and to find locations for mating and raising their young. So we as humans are pretty similar. So for example, we also store objects and we retrieve them at a later date. So we put them in our cabinets or in our fridge, and then we save them for another hour in a day or later on during the week. So we also know the location of many places, such as where we study or where we shop or where we play. And we can also track our location and our starting location so we can technically navigate to and where we came from. So the distinctive nature of many of these spatial abilities suggests that they are mediated by distinctive neural circuits. So we're going to go over four behaviors. So we're going to go over route following, piloting, catching behavior, and dead reckoning. So the simplest behavior is route following. So here you just follow a trail or you're moving towards an object cue. So this is also called cue learning, and I put a little example here with ants. So this is an example of ants following a chemical pheromone trail to get food. So once one ant finds food, it will walk back to its home and it will leave a chemical pheromone trail for others to follow. So in no time, you'll have hundreds of ants on your food. So this is route following. They're following this chemical route. So this also makes sense when in 1 second you have one ant on your food and a couple seconds later you have 100 ants on your food. So another spatial behavior experiment is piloting, and we're going to go over a couple of examples of that. So piloting is the ability to take a course that is not directly marked by a route, and you'd have to learn the course due to spatial cues. So a really good example of this, what is used in a lot of labs, is called the Morris water maze task, and you can see that on the right. But we're going to go over that in more detail in the next 2 slides, I think. Okay, and the third behavior that I wanted to talk about is catching, or catching food. So birds, for example, they catch or they hide their food in different locations for later consumption. So these birds have rich insights into spatial memory. So we're going to go over this in more detail in about 3 slides. And the last one is dead reckoning. And this is a form of navigation that depends on cues generated by animals' own movements. So you know your distance, direction, or speed relative to your starting point. Okay, so let's start off with piloting. So again, piloting is the ability to take a course to a place that is not directly marked by a cue or a route. So the Morris water maze task is one of these tasks. It's a task of spatial abilities. So it requires the animal to learn the location of the submerged hidden platform. So it's right here. So the first thing that would happen is the rat would be put into a pool. And remember, rats are amazing swimmers. So on the first trial, the rat will not be able to see this platform, and let's say it will be put in the pool here. It will swim around randomly, often around the perimeter, and then it will swim around randomly until it finds this hidden platform. Then if you take him out of it, and then the next day if you put him back into this pool, for example now on this side, the spatial cues such as the window and posters will cue the rat where to go. So he'll remember, oh, the platform is kind of towards the window. He will swim almost directly towards this hidden platform. So they found that in rats that have lesions, or if they don't have a hippocampus, this rat will keep randomly swimming to find the hidden platform. So the hippocampus in our brain is needed for spatial memory. So here you have the Morris water maze task. You have a couple of different types of tasks. So these are not these don't have water, they don't contain water. These are mazes. So let's start off with this one. This is the Alton radial arm maze, and here animals can walk from arm to arm and technically follow different routes. So if you put food in one of these specific arms, this maze becomes a route learning task. And since all the arms look similar, the animal must learn the location in relation to the room cues to navigate towards that arm. Okay, here we have a T maze. The T maze and also the Grice box, they're kind of similar. Here, a correct response might consist of learning to go to alternating locations to retrieve food. So we call this an alternation task. So in FIU, we actually do that with rats and also with pigs. So these are also forms of piloting tasks. Okay, so let's move on to caching behavior. So some approaches of studying spatial behavior is observed by natural activity of species. So some birds species have excellent memories for the locations of where they have previously found food, and other birds might have excellent memories for where they have stored or cached their food. So among the birds that scatter food in different locations, the chickadee provides super high insights into spatial memory. Chickadees are one of these birds that cache their food. So chickadees store insects and prey and seeds in little scattered sites, so typically in like moss or holes in a tree bark, etc. So a small number of food items are stored at each site, and cache sites are never reused. So in about a year, they have about 1,000 sites of where they cache their food. So these birds use distal spatial cues rather than social or local landmarks to recall the location of their caches. So distal spatial cues are objects that are some distance away from the cache site, and local cues are close to the cache site. So they use distal cues. So results of studies comparing these birds that cache food with birds that do not cache food indicate that the hippocampus is considerably larger in birds that do cache food. So you can see that right here. You see the size of the hippocampus, or in this case, the forebrain. If they store food, it's way larger than if they do not store food. Okay, so a little fun fact. Hummingbirds actually have the largest hippocampi relative to body size of all birds studied. So this suggests that the hippocampus contributes to their remarkable spatial memory. It's cool, right? Even though these birds are so small, they have the largest hippocampi relative to body size. Okay, moving on to dead reckoning. So dead reckoning is derived from the phrase deduced reckoning. So it is a form of navigation that depends on cues generated by an animal's own movement. So it is the animal's ability to know how far it has traveled and where it is in relation to its starting point. and it can even monitor its speed and travel time and knows when to change directions when necessary. So Charles Darwin was the first to suggest that animals could use dead reckoning to navigate. So humans use dead reckoning too. However, we often use little tools to do this. So it was technically an early form of navigation by sailors. So Columbus used it on his journeys between Europe and Central America. They used, for example, a compass to monitor direction, and a sailor could calculate speed by throwing a piece of wood overboard of a specific density, and then the sailor would chant until the wood has passed a certain point, and the length of that chant would correspond to a specific speed. Funny, huh? Now we have all these computerized things to tell us all these things. However, non-human animals, they use their self-movement cues to do so. So they can use their sensory flow or movement commands, and they don't have a compass or a speed measure. So they have to do it internally. So non-human animals have to get this information from their self-movement cues, and they can return home even when all auditory and olfactory cues are removed. So they don't have to rely on olfactory and auditory cues, even though it does make it easier. So studies have shown that the hippocampus is also necessary for dead reckoning. So damage to the hippocampus will disrupt dead reckoning. So here on the left, you see a ship that starts from a known location, and when it arrives at a destination, it can return to a starting point without using external cues, just by calculating time, direction, and speed. And on the right, you see a cute little rat foraging for food. So he'll make a number of turns and stops to try to find food. And when he finds the food, he is able to go straight home without having to follow the entire path that he came from. So the rat does not have a clock or a compass or a speed meter. It uses internal processes for dead reckoning, so he can go right back to his home. Okay, so let's go over some hippocampus functions. So the hippocampus is really important for spatial navigation. Specifically, birds that catch food have larger hippocampus. Without the hippocampus, rats will not learn to swim directly to a platform after learning where it is once. They also have impaired spatial memory, and the hippocampus is important for dead reckoning in rats. So why is this hippocampus structure so special, right? So in the next video, we are going to talk in more detail about this, but I want to give you a little hint. The hippocampus has special cells. So specifically, it has 3 types of cells that we're going to cover in detail in the next video. It has place cells, head direction cells, and grid cells. So place cells tell us in what location we are. Head direction cells tells us in what direction we are facing. And grid cells will tell us where we are in an area. Okay, so this was the end of this video, and I will see you guys back soon in the next video where we talk about these cool special hippocampal cells.